In this video, we'll look at the editing options available in SeqMan Pro for .sqd project files, including trimming and untrimming individual sequences and groups of sequences, removing conflicts or gaps from the consensus, direct editing of aligned sequences, alignment adjustments, and finally, exporting the edited consensus and aligned reads. Note that these editing options are not available for BAM files. Sequences are often trimmed by SeqMan Engine or SeqMan Pro during assembly. You can tell a sequence has been trimmed when you see an arrow at the end of the sequence. To restore the trimmed portion of the read, simply drag the arrow away from the middle of the sequence. Previously trimmed sequence is shown with a yellow background. Similarly, you can manually trim sequence ends by dragging the endpoint in toward the middle of the sequence. You can also trim multiple sequences at once by selecting the end portion from the consensus that you want to trim, and then selecting Sequence, Trim Ends. SeqMan Pro also lets you type edits directly into the consensus. For example, you could remove a gap by selecting it and then hitting the backspace key. You can also type bases in. Note that when you make an edit to the consensus, all of the aligned sequences are edited in the same position. Similarly, you can type directly within a sequence to make edits. For example, if I want to delete these two bases and gap, I can select them and hit the backspace key. By making that edit, I've now adjusted the alignment of the sequence. To correct this, I'm going to use the sequence sliding tool and then shift the entire sequence to the right. Once you have completed any necessary edits to your alignment, you may wish to export the consensus sequence. To do this, go to Config, Save Consensus, Single File. Here you can name your file and specify the file type. You can also choose to include gaps in your exported consensus. You may also export the aligned sequences in a config by going to Config, Export Sequences, and then selecting single or multiple files. Again, you can choose to include gaps in your exported sequence, and you can also choose to export the entire sequences, including the trimmed portion of the reads. By default, the trimmed portion is not included in the export. If you have further questions about working with SeqMan Pro, or to request a fully functional free trial of our software, please visit our website, www.dnastar.com, or contact us at support@dnastar.com.